Happy to, I guess, end this session a bit late, so I'll try to keep on time. I'm also, as a disclosure, I'm the chief scientist of Cytoreason, a company I co-founded. I'm also the co-CSO of the Human Immunome Project, which is a nonprofit related to mapping the human immunome across the globe, and happy to follow up on this. And so really I'm going to try and give you a kind of an angle from the immune side. This was mentioned in today's session several times. Here's a bit of data just to kind of give you some reflection. On the left-hand side, you can see your standard white blood cell count. This has really been measured in humans since 1957, so it's been around, and it particularly tells you everything we know on kind of the immune system from blood at a resolution that if you were to be an immunologist, you would say is totally irrelevant. And that's because really for each one of those different cell types that you see here, we now know there are hundreds of different cell types that can be defined, each with its own function, and those ultimately relate to maintaining health and disease. And just to give you a sense of those, so if you think about every cell as having the abundance being a reflection of its your ability to function in some way, here's a blow up of percent lymphocytes, and everyone here is a physician knows this is quite common to see a lot of variation in this. And so here's some data. Every person here is a yellow line, and you can see there's a huge amount of variation between us. If we were to profile each one in the room, would look different. And there's a change that happens with age. And this isn't just at the resolution of percent lymphocytes. If you zoom into say percent CD8 T cells or subsets of CD8 T cells, you will see the same thing. A huge amount of variation between individuals, as well as remarkable changes that happens with aging. And so it's a complex system, and one would maybe expect this because this is a sensory system. The entire idea of the system that it senses changes within external to us and within us, and it needs to respond, and so it keeps changing all the time. And give its function and role in health, actually understanding it and how those changes are happening is key. And so the reason would be ultimately, as I say, if imagine somebody walked into a doctor and said, "Hey doc, how's my immune system doing?" We want to have a good answer for this, and the resolution that the immune system actually is and the resolution we measure in the clinic are so remarkably different. I would argue we don't have a good understanding of this, and there's a lot to be understood. And so with that, here's a way of thinking about this as a thought experiment. Some people here may know the Waddington landscape from the 1940s, where on the top of this graph you had a stem cell and genes that were ultimately related to differentiating this cell. So from an immune perspective, I'll say rather than thinking about a single stem cell, think about what you see here as a composition of all these different immune system cells, each of which we have different frequencies for, and those changes as we live life, right? So it's almost not just genes, it's your life history, just being exposed to life, and your frequency will change, and maybe this is the frequency that you have in one particular place in the globe, but another, and maybe this would be disease, but really it's about living life. But if I wanted to think, so age here is our differentiation, the equivalent of the stem cell. But if I actually wanted to think about this from the immune perspective and the path that we take, I'd say the prime path we move on is what I would call immune aging. So how does the immune system change with age? And so some take-homes from from my talk would be that what we've identified is this is a pattern change. This is not random. There's a developmental process by which we all change. We are now able to quantify it, and we keep improving in doing so. People travel this path here at different rates. Each one of us moves at a different rate on it, and it is such a huge change in healthy individuals. This is actually what I would call the first principal component of immune system variation in human, where basically this is the biggest difference that we would see among healthy individuals, and it matters clinically. So basically, if you think about clinical medicine and its relationship of the immune system to it, which I would say is quite critical, your clinical phenotype sits below, above the immune system, and the immune system plays a role, and where your state is on the system is ultimately going to matter. So to really quantify this, back in 2007, we started a study with Mark Davis. I was a postdoc with Mark. Mark had a great idea. Let's start following humans and measure the immune system in high resolution. In 2007, this was science fiction. The tools to do so were really not yet there, and we've been developing them along the way. And we now have a study that's been running for 17 years, where we do longitudinal immune monitoring at high resolution in humans. And in fact, this is, to the best of my knowledge, the longest running high resolution study of the immune system. And I'm just going to show you a few slides from a paper we published in Nature Medicine in 2019 that was led by two students from my lab, Ayelet Alpert, who's an MD PhD, and Shai who's a postdoc, where we describe the changes that are happening in 9 years over the 150 people that are participating in this study. And given there's a lot to show, I'll show you really a few plots. This is just looking at the older individuals, 60 and above, in the study, and a subset of the individuals that we've removed any technical differences on measurements from one day to the other. And the y-axis shows you naive CD8 T cell frequency. The reason I'm showing this is because naive CD8 T cells are one of the hallmarks of the aging immune system, where your thymus shrinks and your naive CD8 T cells drop down. And it's been reported hundreds of times that naive CD8 T cells drop with age, and this is the black line. See, that's the average in the population. But we have enough data to model how this behaves in every individual differently. And so that's the colored lines. And you can see the differences in rates between individuals. And in fact, some people actually go against the dogma. Their naive CD8 T cells go up in those years of the study, and it matters. So this is just one cell type like this, but actually we have many different cells, but we're seeing differences in the rates of change between individuals, and in fact that the average really doesn't describe necessarily characteristic of what is happening. In fact, we have so many different cells that we can describe individuals not based on, say, their height or weight and so forth, but really what their immune system looks like at high resolution. So imagine many, many different individuals, and for each we have a vector of many different immune cell types, and we can take this vector and basically try and mesh them together, or tie them together to describe how does immune aging happen throughout the entire lifespan, or at least from young adulthood to older individuals. So build a trajectory that estimates the immune aging process that we go, trying to capture the developmental process of immune aging. And this is what we did, and you can see this on the left-hand side. This is a graph that's basically computed by an algorithm that does dimensionality reduction. The input to this figure is only for every person what their immune cell types are at high resolution. In fact, 18 different cells per individuals were entered in, and not what the age of the person is, not what year they came in or visit of the study. Nothing else, just your immune system. And the graph here, every point is one person. They're colored by their chronological age. So on the right-hand side, you see people who are young chronologically, and their immune system kind of looks young. This is the heat map describing them on average. Young people are kind of boring, so I don't spend a lot of time talking about them. And on the left-hand side, you can see as you move on this dimensionality reduced figure, right? So think about this like a PCA. You see the individuals, the chronological age breaks down. So there's a lot of noise in what chronological age is. And in fact, there's disagreement between, I would say, actually a positive noisy correlation between chronological age and what is changing in your immune system, right? So this pseudo time, we can describe individuals and how they change on it. This is how people are organized on this. And you can see the main cells that are actually driving this, and they're changing over time. And just to give you one example, this is one person, the red arrow here, as he shifts in 4 years, right? So from this profile, the color denotes the abundance of the cell scaled, to 4 years later, totally different, okay? And for each of us, this is the types of things they're doing. And as I said, one of the really interesting things here is this is conserved. Despite the fact that if I queried here, we would all look very different, basically we all travel the same non-branched trajectory, this conserved developmental pathway, just at different rates. So how do I see this? Well, here's the old individuals in the study and the young ones. The young, you can see, are kind of noisy. They move around from the longitudinal data. So on the trajectory, the pink are the older individuals. Their slope is always positive. They keep moving forward on the trajectory, okay? Just at different rates. Here's one person moving slowly. Here's another one moving much faster. And in fact, as they move on this developmental trajectory, as your immune system changes, we now test them functionally. So we throw cytokines at the immune cells and we say, how are these cells responding? And bad news, possibly, As the more you move on the trajectory, the less your cells are responding, the more attenuated your immune system is, right? And so you can think about this, maybe it's a good question if it's a good thing or a bad thing, because it could be that in some places around the world, actually having an attenuated immune system may be a good thing for you, and maybe that's how we've evolved. And so we did an analysis of a data set that was published from Mark Davis's lab, looking at Bangladeshi kids versus children in Palo Alto. You can see the children here, three-year-old children in Palo Alto, and older adults in Palo Alto, young individuals who are about 30 years old. And you can see a marked difference between the Bangladeshi kids and children in Palo Alto. In fact, they're coming close to these individuals with respect to describing what their position is on the trajectory, right? So basically, as you think about it, we can now give you a number, draw a blood sample, measure 18 cells, and give you a number of what that describes what your immune age is. We call this image, and this is not just about aging, it's really about being exposed to life. So you can think about this almost like a road that we all travel on, right? As I said, we can measure this now actually by several different data modalities and quite cheaply. Eric Feldman, who's a student in the lab, is sitting here, and he's developed a flow cytometer test for this. And the image score is basically where you sit on the trajectory. Now, one question you would think, given the immune system, is that if we're all traveling on one road, just at different rates, what if I took an off-ramp? What does it matter if, like, say, you vaccinated me, or what does it matter for clinical medicine where your immune system is like? And so we looked at this, right? Because it's important to understand if everybody's going in the same way or not. And so the first thing to look at is something big, like survival. And so we developed a gene expression score that basically predicts your image score, your immune age, on where you sit on this trajectory. And because our study is small, we moved from this 150-person study to taking on the Framingham Heart Study, where if you actually take the individuals in the Framingham Heart Study and you score them for immune aging, you can see the differences in survival and also in cardiovascular risk. This is part of the published paper. And then if you now add in all the clinical factors, you can see that and you model image. So for those who are not familiar with Framingham Heart Study, this is the longest running kind of cardiovascular study. And a lot of what we know about cardiovascular medicine comes from this. There's a lot of clinical information we can model. We model this, and we modeled their immune age because they all measured blood gene expression, and so we can estimate it. And this is a significant survival association, and the odds ratio is 1.05. In fact, in the Framingham Heart Study, they also measured methylation, just like Steve was talking about in the previous talk. And so here's just a plot where a table where we take in a lot of different clinical factors and the different kind of published methylation clocks. And what you can see is that immune aging is always a significant factor, even when the methylation clocks are modeled as part of the Cox model. And in fact, often it's greater, but most important is that it always adds additional information. So kind of suggesting that we're not capturing everything that we need still with the methylation biological clocks. And in fact, when we did an analysis, we developed a methylation-based clock for immune aging, and it does not overlap with the CpG sites that are coming on from the published clocks, suggesting there's additional information that can be captured from this. Another way of kind of looking at the same direction is so important for survival, cardiovascular risk, but let's talk about something much finer grained. If we're talking about the immune age, well, what's really important are immunotherapies. So we ran a study that was led by Mayan Briller, a PhD student in the lab, where we looked at estimating immune age, how do individuals respond to COVID vaccine mRNA, the Pfizer vaccine, where the study was designed to take individuals across many different ages and estimate their immune aging. This is a collaboration with Yuval Doh and Dana Wolf from Hebrew University and Hadassah. And here's just kind of two plots from the study. On the left-hand side, you can see there are neutralizing antibodies for COVID, for the COVID vaccine. And when we model it with chronological age, or when we model it with immune age predicts neutralizing antibodies much better than your chronological age. In fact, if we divide the data into immune age, young or old, or chronological age, young or not, old, you see significance in immune aging not captured by chronological age. And in this particular data set, immune aging explains 20% of the variation in the data, whereas chronological age explains 10. There's still a lot for us to do work on in understanding immune variation. Another interesting thing from the study is that when we try to predict neutralizing antibodies, we get a bunch of features that predict it. And you can see those on the left-hand side for positive features and also for negative ones here in blue. But if you do this in the older individuals in the study, the features are not the same. So they're no longer predicted. The same features that are used to predict the immune system components that relate to how you will respond in terms of building neutralizing antibodies for the vaccine seem to not play a role in the elderly. And we took this finding and went with it to a much larger study looking at over 600 individuals with flu responses. You can see a heat map here where where the individuals are organized based on their immune aging score. So this is the columns, and the features that correlate with your neutralizing, with your flu responses are listed in the rows. And what you can see is basically the immune system, and what's important in different pieces of like your developmental process actually changes what those features are. Just like if we think about, say, gender differences, how biology in men and women works would be different. It looks like in aging, that's the same thing. What is important in young to predict responses is not really what plays a role in when we're vaccinating individuals, older individuals, and we are hoping for them to respond. And of course, we have a problem because older people do not respond to vaccines and young do, and we develop the vaccines in the young. Kind of led me to this analogy where I think about the immune system of older individuals, and I think, no, it doesn't. It's not that it doesn't work, right? It's that they're playing a whole different strategy to actually score a goal, right? So if you were thinking about, say, soccer players, young soccer players that run around for the goal, the older individuals, if they chose to play soccer, they would play a whole different strategy. You'd still have a goal. Maybe they miss sometimes, but they have a whole different strategy. So thinking about the immune system as being a conserved thing where it's such a dynamic process is actually probably not a good thing, and we need to rethink how we do so. So I'll end this part and say, in summary, that what I hope to show you in this brief talk is that immune aging follows a pattern trajectory of change. We travel at different rates on it, and it's the first principal component of the system. We can basically map you now to a reference. We can do this with cell frequencies in cytof and flow cytometry by gene expression in whole blood and PBMC by methylation, and proteomics is coming. It has molecular and chemical facts, and it's basically a lifelong pathway to study and manipulate. And with this in mind, I will put a kind of brief ad to where, my next slides are missing. Here it is, okay. So we've basically decided to put together the Technion, a Healthy Aging Institute. And I was gonna say it relates, from our perspective, the immune system, the big thing is how it relates to physiology. I direct this institute. We're just launching now. It's called iTech Age, is the kind of the think tank around it, which we'll partner with a lot of different groups. It has a lifespan cohort of over 1,000 individuals that we're tracking, a systems medicine center for kind of bringing in kind of a lot of the different types of multi-omic measurements. And really principal to this is over 60 researchers at the Technion who've all kind of been joining into this established groups and themes. The lifespan cohort is a shared Technion resource for all of them to study, and they span from looking at cellular mechanisms of aging and systems aging and immunology and endocrinology to brain and cognition, biomarkers of aging, personalizing foods and drugs, assistive technology, and ultimately even looking at the communities where we have people from the Faculty of Architecture where we'll be doing those types of research. So thank you, and this is the lab. It's all really done by the students. Thank you. I guess as the chair, questions from the audience. Hi, Gil.
Okay, my question to you is the following. You said that the immunologic aging and the what's so-called the cytosine clock are not best fit My question, and the other thing that you said that there is different velocity or different fitness between, age people that fit to immunological aging. My question though, are those that fit better to the immunologic aging are also, how do you say, can predicted by the biological clock?
You mean by epigenetic clocks? The short
Correct.
answer is we don't have data for this because I don't have data sets that have two methylation time points and two immunological time points. I actually, as you asked this, I realized I can actually generate that data. So I will let you know next time.
Hi. Very interesting indeed. Did you thought about what factors actually influence the immune system? Because let's say, for example, a person that immigrate from one country to another and there was a climate change and food change and everything. So this is external influence on the immune system. So those kind of people, do they have better immune system or does it hurt the immune system and they have less?
It's a wonderful question. I think because it's a sensory system, there's many such fascinating ones. I keep thinking about the migrating ones. I don't know. I will say I don't think we even understand. So certainly, larger immune aging seems to predict cardiovascular risk. We know we can manipulate it. Like people who respond to inflammatory drugs drop in their immune age clock for a few kind of the immune aging process. But I'll give you an example. So for cardiovascular disease, we know high immune aging is bad. Bone marrow transplant, which is a study that actually Eric is running here. When we give somebody gets a donation, do you want to have a high or low immune aging? We don't understand yet. Certainly, data that has looked. There's people who've looked at the change that happens when you shift from a rural environment to an urban one. And there's a huge immunological change relating also to your diet. And we haven't mapped that data, but if I were to kind of estimate, I would say that you're probably getting an increase in immune age when you see this. There's also increased incidence, of course, of chronic diseases, but we have a lot more to do.
Thank you.
Thank you. 30 minutes late, I end this session. I guess, Ilya, we're taking still a break, right? Well-deserved one.